Okay, so I think we can start right now. So please, for all the participants, can you turn off your mic and video? Okay, so good morning. And today we have a quiz lecture from USA, and I will be conducted by our quiz lecturer, Mr. Ramesh Subia, and he is currently a postdoc fellow at School of Dentistry, Oregon Health and Science University in USA. And I'm gonna be your host and moderator. So today uh, we are to be honored that we have Mr. Ramesh as our guest lecturer. So he will present about the microenvironment determines cell fate. So I think this topic is related with our major, especially in tissue engineering. And before he start his lecture, I would like to introduce shortly his curriculum vitae. So I'm gonna read his CV. So he obtained his bachelor degree from the wait a minute from the Dr. MGR Medical University, Chennai, Tamil Nadu, India, and with his research about synthesis, characterization, and antimicrobial studies of some 1,2,4-triazole derivatives. And, and then he obtained his master degree from Gachon University, South Korea, with his research is about the fabrication of bio-nanofilm for biomedical applications. And then the last, he obtained his PhD in biomedical engineering field from Korea University of Science and Technology, UST, and Korea Institute of Science and Technology, Seoul, South Korea. And currently, he is a postdoctoral fellow at the Oregon Health and Science University. Please, for other participants, could you mute your mic? Uh, so I'm gonna repeat for the one. So currently, he's a postdoctoral fellow at Oregon Health and Science University with the project Microengineering Vascularized and Innervated Bone-like Scaffolds as an alternative to autologous bone graft. So his main field is about the tissue engineering, and today this lecture is attended by students from the first year and the second year, and I think several from the third and fourth year, and also another lecture. Okay, so I think we can start right now for you, for Mr. Ramesh to present.
All right. Yeah. Thank you, Dita, for the wonderful introduction. Let me begin now. So I'll start my screen sharing to make sure it is visible by the students.
Yes.
It's fine now.
Yeah. Wait a minute. I want to inform for the student. If you have any question, you
Uh-huh.
can raise your hand or you can write on the chat box. So we uh, we can discuss about it after Mr. Ram's presentation. Thank you.
All right. Yeah, thank you. So, Salamat Bagi students. Myself, Ramesh Subaya, I am a postdoctoral fellow working at Betasoni Lab in Oregon Health and Science University, which is in Portland of Oregon State, in the northwest region of the United States of America. Let us discuss about microenvironments that determine stem cell fate. So, when we talk about microenvironment, it's like a small story I would like to say. So whenever we live in our own house, we feel happy. And if we live in a different house, we feel strange. We know, oh, this is not our house. So microenvironment, it's something like that. Microenvironment is like a house for stem cells, where stem cells feel happy and then they can do whatever they want. So that is what I'm going to discuss today. Before I begin, let us discuss about human body. As we all know, human body consists of trillions of cells that collectively build organs with systematically biological, physiological functions. Active function of organs depend on cells. How? By consuming nutrients and converting them into energy. So every cell types are unique in exhibiting their own functions. Constantly, cells undergo several mechanisms to self-regulate and to be functionally active. For an example, insulin is produced by beta cells. Nobody comments when to, they know when to secrete insulin. So that's what it means. But any malfunction of cells leads to organ failure and cause of death if untreated. The number of organ failure is in an increasing order every year. Besides, severe injuries and chronic diseases such as heart failure, congestive heart failure, that makes up to 31% of all global deaths. Spinal cord injury, liver cirrhosis, then renal failure, and diabetes mellitus, which is about 211 million patients worldwide suffering from diabetes mellitus. All these severe chronic diseases are resulting in harmful life. Reconstructive surgery, as a result, have emerged during the last century to save the life by transplantation using either isograft, autograft, allograft, or xenograft. In most of the cases, patients' immune system should be suppressed to prevent rejection of the graft. About 666,000 of transplantation have been performed during the year between 1988 till 2016. Liver, kidney, liver, heart, and lung transplantation, they top the list of the transplantation. But the major limitation with transplantation related reconstructive surgery is the availability of organ donors, graft accessibility, donor site morbidity, prolonged operation time, and cost. So we need an alternative strategy. We need an alternative treatment, a field which is considered, which is tissue engineering. So tissue engineering is a rapidly emerging biomedical research field, provides an alternative to grafting and transplantation, and also reserves true potential to confront the transplantation crisis caused by the shortage of donors, tissues, and organs. Then let's discuss about tissue engineering. Since last decade, the field of tissue engineering and regenerative medicine has received much attention for the proven effects on tissue replacement, repair, and regeneration using combination of three elements, which is biomolecules and then scaffolds and stem cells. I mean, biomolecule agents, stem cells, and biomaterials. So importance of biomaterials, scaffolds, space and nutrient supply at the regenerative site, delivery system for biosignaling molecules, in vitro cell manipulation, and engineering biological function of cells. These are the key factors that is considered important in tissue regeneration, as we have seen in this figure. So we have to know how to engineer cells, such as we should be aware of cell source, what kind of cells they are, iPSCs or adult stem cells. And then we have even developed genetic tools to modify the cell genetic content, such as using CRISPR technique And then we have engineering materials like novel chemistries have been developed to tune the material properties. And then we have growth factors, and then biomechanical cues, and then finally latest technologies such as 3D printing, self-assembled molecules, and decellularized organs. All this together, tissue with the help of development in all individual field, tissue engineering has been very fruitful over the decade. And then today we will discuss about each and everything. And then the most important thing is tissue engineering is not just one field. It coalesces the fields of cell biology, biochemistry, molecular biology with material science, chemical, and bioengineering. Moreover, advances in understanding these strategies, such as biophysical cues on cellular behavior and tissue architecture technologies such as decellularization technique and 3D printing, have contributed to engineering tissues of considerably improved structural, compositional, and functional resemblance to their native counterparts. That means we can almost nearly synthesize a bone that is close to what is found in our body, what native microenvironment. That is what it means. And then the foremost important part of tissue engineering is stem cells. So cells are at the forefront in regeneration field, particularly stem cells, so which are a class of immature and undifferentiated precursor cells that are able to self-renew and differentiate into specialized cell types. The most popular stem cells, when we say that, comes embryonic stem cells. Embryonic stem cells are isolated from the inner mass of the blastocyst. It is totipotential. Totipotential means you can make literally any organ from embryonic stem cells. But the problem with embryonic stem cells, it is prohibited. Use of embryonic stem cells is prohibited because it's immunogenic, and the rejection is a problem, and moreover, ethical problem. It's like killing a fetus. So use of embryonic stem cells in the research field is prohibited. In contrast, adult stem cells has been evolved, such as hematopoietic and mesenchymal cells found in the mature tissue, like for an example, bone marrow. They have been super popular over the decade because they are capable of generating multiple lineage of cells, such as lung, bone, or blood cells, chondrogenic cells, and as well as muscle cells. So they have been like very fruitful. Recent clinical results have demonstrated the feasibility of stem cell therapy in tissue repair. However, the process a stem cell undergo to be differentiated involves thousands of questions which are still open and has to be resolved for future tissue engineering research. Particularly, what are the factors controlling the stem cell differentiation that we call microenvironmental factors that I explained in the beginning of the lecture, our house. So our house, we live in our house, how comfortable we are. So similarly, stem cells live in a microenvironment that design their behavior and differentiation, which we will discuss one by one in the following slides. When we talk about cellular microenvironments, researchers have found all this, whatever we see in the figure, all these cues, researchers have demonstrated each and individual cues play an important role. So cellular microenvironment design the behavior and differentiation of stem cells. That includes several factors, particularly biochemical cues. When we say biochemical cues, that is growth factors or small molecule or cytokines. That means cells secrete certain protein for their own purpose. And then we have biophysical cues. When we say biophysical cues, we have mechanical strain, shear stress, topography, dimensionality, and stiffness and elasticity. Finally, extracellular matrices. Among all these major elements, whatever I said now, growth factors, stiffness, extracellular matrices play a very, very important role in tissue regeneration. So engineering these cues whatever the said cues is vital in controlling the differentiation of stem cells, particularly delivery of soluble chemical stimuli such as growth factor using fabricated biomaterial architecture and extracellular matrices with tunable mechanics. Let's discuss about biophysical cue. What is biophysical cue? So if we look at our own body, the mechanical property of our organs vary is differed from organ to organ. For an example, bone is the most stiffest. Bone is the most stiffest one, which is almost like 50 from 10 to 50 kilopascal, depends on whether it's cancellous bone or long bone or a flat bone. Whereas in case of muscle, which is around 10 kilopascal, brain is the softest, which is less than 1 kilopascal. Fat is somewhere in between like 2 to 3 kilopascal. So our own body organ mechanical property differ from organ to organ. So there was a study in 2006 by Disher group. It's considered a breakthrough study, the first study that demonstrated when stem cells cultured on a different stiffness material, different stiffness substrate, the mechanical property played an important role in discerning their differentiation. For an example, the substrate they used from 0.1 to 1 kilopascal, less than a kilopascal, stem cells differentiated to neurogenic. And then from 8 to 17 kilopascal, stem cells differentiated to muscle cell. Above 25 till 40 kilopascal, it differentiated into bone cell osteogenic cells. So it's a breakthrough study. The first biophysical cue explained experimentally that stiffness has an important role on stem cell differentiation. Since then, many material science have evolved, developing materials both naturally derived and synthetically prepared to tune the stiffness controllability. So in natural biomaterial, we have collagen, gelatin, alginate, and then hyaluronic acid, most popular one. And in synthetic biomaterials, we have PDMS, polydimethyl methylsiloxane, then polyacrylamide, then we also have polyethylene glycol. This particular biomaterial section, at the end of the lecture, I will describe one by one the advantage of those biomaterials. So in order to evaluate biophysical cues, our body microenvironment is completely different than these materials, which we call polymer or the natural biomaterial. So they lack the environmental cues stem cell encounter in our body. This material does not recapitulate, does not mimic them or don't provide them. So we need a material that is very, very closely mimic the native extracellular matrices, for which what we have done, we try to isolate, we developed a method to isolate extracellular matrices from the cells. So when we talk about extracellular matrices, cells upon culturing, they adhere and then they start to produce proteins in the forms of matrices underneath wherever they are cultured on. That is called extracellular matrices. It is a well-organized three-dimensional network composed of variety of proteins, proteoglycans, and other biomolecule components such as fibronectin, laminin, vitronectin, and many others. So the complex and hierarchical ECM structure provides a microenvironment that closely mimics native microenvironment. So the previous study, Disher group, they published stiffness dependent stem cell differentiation. So we have developed an idea of changing or tuning the material property by cross-link. What we did, we cultured fibroblast cells on a substrate. Upon establishing ECM secretion, we decellularized them to isolate only the pure decellularized matrices. These decellularized matrices can be cross-linked with genipin, which is a cross-linker. Genipin is a wonderful cross-linker compared to EDC, glutaraldehyde, and formaldehyde, because it's not only effective, also safer, very, very safe, less cytotoxicity than glutaraldehyde and EDC-based cross-linking. On top of that, genipin achieves intra and inter cross-linking of protein. So that means your cross-linking is very, very high. and that can be controlled by the concentration of the crosslinker that you use. And the beauty of this material, this crosslinker, is you have native freshly derived cellular matrices, ECM from the cells, and then you add 0.1%, 0.5, 1%, 2% of genipin to crosslink. And what happens? The structure remains the same, the protein remains the same, but the mechanical property is increasing upon cross-linking from 100 Pascal to 20 kilopascal. What the graph shows here, from 100 Pascal to 10 kilopascal, when the concentration kept between 0 till 2% of the 2% weight by volume of the cross-linking agent. Then this biochemical cue is very, very tunable. When we say tunable, that means you can control the stiffness of the extracellular matrices. And this ECM, tunable ECM, is capable of differentiating stem cells in a stiffness dependent manner. So we have soft matrices. When you culture mesenchymal stem cells on soft matrices, that predominantly direct the differentiation of stem cells towards adipogenesis. In case of stiff matrices, that predominantly direct the stem cells towards osteogenesis and chondrogenesis, as confirmed by oil Redwood staining here on the right side, oil on the top, oil Redwood staining, then safranin O staining, and alizarin red stain. This is also confirmed by gene expression for PPARG and then collagen 1, collagen 2, aggrecan, annex 2, and osteocalcin. So from this study, we proved that yes, ECM can be obtained from the cells, which is a very native source, and then the tunable mechanical property can be tunable, and stem cell differentiation matches with whatever reported in the past. So this material can be like a potential replacement for the native, naturally derived or synthetic biomaterials. And then we talked about an external mechanical stimuli on stem cell differentiation. Stem cells also undergo biophysical and biochemical changes during the process of differentiation. For an example, osteogenic progenitor cells, when they undergo differentiation towards osteoblast cell, what will happen? Type 1 collagen, osteocalcin, osteopondin, and bone sialo protein, all these protein will be highly expressed upon during the differentiation period. Not only that, the later stage, the matrix will be mineralized. Upon mineralization, the stiffness of the matrices increases, and then which we confirmed by the mechanical property of the differentiating cells. From what we have found, from almost like 10 kilopascal to 50 kilopascal, upon from day by day, the differentiating cells mechanical property increases significantly. So this is like a proof that in our bone, when stem cells differentiate into osteoblast, the mechanical property increases. One of the important tool to measure the mechanical property of organs or tissue or cells, it has to be performed in wet condition. And then when we say mechanical property analysis in a wet condition, powerful tool called atomic force microscopy with a nano indentation technique is highly useful. There are several other papers discuss about the use of atomic force microscopy, which you can refer later. It's a very big field. And then now we have discussed about biochemical, the mechanical stiffness on stem cell differentiation, how cells changes their stiffness mechanical properties, and then can we use an external force to control cell stiffness? Yes, for an example, magnetic force. So when we say magnetic force, if we bring cells under the magnetic field, what magnetic force will do to the cell stiffness? This is what I wanted to explore. So for this, I prepared magnetic nanoparticle, and then magnetic nanoparticle is it exhibits cytotoxicity. So to render biocompatibility to the magnetic nanoparticle, we surface functionalized with biopolymers such as PLGA, polylactic glycolic acid, and then alginate using electrospraying or emulsification method. This method I will discuss later in the lecture. So by functionalization, we can render the biocompatibility of the magnetic nanoparticle, and then magnetic nanoparticle and then functionalized nanoparticle retain the same structure and also the size. Not much differences in their magnetic property. So using this, when we treat the cells with magnetic nanoparticle and then PLGA coated or alginate functionalized magnetic nanoparticle, when we apply magnetic field, we can control cellular uptake of the cells, cellular uptake of magnetic particle. So an applied magnetic field promotes intracellular uptake irrespective of whatever the magnetic nanoparticle. So upon uptaking, what happens? The surface roughness increases and then reduce cell height and spreading area. The stiffness of the cells also reduced in magnetic nanoparticle because of its cytotoxicity. Whereas PLG and alginate functionalized magnetic nanoparticle, they increase the stiffness without any cytotoxicity. So magnetic nanoparticles potential to be used for targeting cells, tissues with magnetic forces, and other biomedical applications such as cellular imaging, magnetic hyperthermia, and most importantly to manipulate cell stiffness for differentiation studies. This is a general introduction about biophysical use such as mechanical property, how mechanical property affects cell behavior, and what are the changes cells undergo during the differentiation process, and the external force, what are the external forces, how we can apply that to change the mechanical property of the cells. This is what we discussed. Let's move on to the next cue or factor, microenvironmental cue, which is biochemical cues. And when we say biomolecules, so biomolecules are secreted by cells, right? So they are secreted by cells and they influence cell signaling pathways. So they come and they bring information to the cells like what are the functions they should exhibit. These cell signaling pathways influenced by biomolecules such as proteins or growth factor or cytokines through extracellular and intracellular mechanism. And their delivery have found various tissue engineering applications such as bone regeneration, blood vessel regeneration, cartilage, and other organs. So we need to identify what are the biomolecules that causes this differentiation, how they are how they are helpful for particular tissue engineering. So we need to identify that. Before discuss about growth factors, let's understand what is happening in our body. So cells, there are 4 types of signaling happens in our body. Autocrine. So for a cell secretes something for its own function. So it secretes and it takes. And then we have signaling across gap junctions. Cells connected to another cells through gap junctions. So one cell secrete biomolecule, a protein or cytokine, and transfer to the next cell, adjacent cell through gap junction or different axis. Then paracrine, neighboring cells. A cell little far cell secretes some molecule to be targeting the cells. Last one is endocrine. That means cell secretes something for the far cells. So this molecule enters bloodstream and when it will take a while to reach the target cell. So in a way we can say whenever we have an injury, for an example, skin injury, what happens? The entire body actually react to heal the injury. So many cells participates, they secrete different biomolecule, the neighboring cells or adjacent cells or far cells. All these biomolecules will come to the injury site to promote regeneration or healing. So biomolecules, when we say biomolecules, simply they are called growth factors. They are soluble proteins that actively participate in cell signaling pathways and also regulate cell behavior. a number of growth factors have been recognized along with their specific transmembrane receptor ever since the first growth factor was discovered by Dr. Rita. Nerve growth factor and epidermal growth factor are the first two growth factors discovered, and they won Nobel Prize for that. Let's see type of growth factors. And the first and biggest family in the growth factor family is TGF beta. That consists almost 33 members in TGF beta, including BMPs, bone morphogenetic protein, growth differentiation factor GDF, GD, and the second biggest family growth factor is FGF, which is fibroblast growth factor. That consists of 23 members, FGF1, 2, 3, 4 until 23. Then we have epidermal growth factor, insulin-like growth factor, or hepatocyte growth factors, PDGF, VEGF, lastly, neurotrophic factors. All these growth factors are important for specific function of the cells. So if one of the growth factor is muted or genetically manipulated in our body, that particular growth factor will not be secreted, will affect in severe problems. So when we say naturally growth factors exist, so many functions. The important functions of growth factors are regulating cell proliferation, self-renewal, migration, and differentiation. So growth factors binding to a cascade of intracellular signaling events through transmembrane receptor that culminate in both transcription independent and transcription dependent changes in target cell behavior. Studies demonstrated that individual growth factor has positive effects on the differentiation of stem cells. For an example, IGF-1 differentiate into beta cells. BMP2 and BMP7, bone morphogenetic protein 217, has play an important role, significant role on the differentiation of osteogenic cells from mesenchymal stem cells. TGF beta 1 and beta 3 on chondrogenic cells. VEGF both on endothelial as well as smooth muscle cells. And all the other things, PMP4 on lung progenitor cells. So growth factors are important for differentiation of stem cells. But when we say growth factors are stem cells, there are other factors affect regeneration. So in case of growth factors, biomaterials and delivery systems, so many small properties can affect the outcome of a particular study. So we need to have a fundamental understanding about each of these factors affecting tissue regeneration. So from the perspective of, in case of growth factor, a biomolecule delivery, we should be aware of what is the dose required. If we give a high dose, it may end up causing side effects such as inflammation, cancer, or heterotopic ossification. These are the biggest problems. So we should be aware of, we should be knowing what would be the required dose in terms of growth factor. When it comes to biomaterials, we should be aware of their molecular weight, solubility. What is the solubility of the biomaterial? Is it cytotoxic or not? The surface charge of the biomaterial, is it degradable? If it is not degradable, it will remain in the body forever. So how we are going to remove that? And then these are the properties in terms of biomaterial. Then we discuss about delivery systems or scaffold. So we need a material for tissue engineering application to deliver growth factor or cells. We need scaffold. So these scaffold will be implanted ultimately at the injured site. So we have to be aware of what size is required, what should be the shape, and then the porosity, diffusion. If it is not diffusive enough, cells inside the scaffold will die. So it should be diffusive enough so that blood vessels can integrate into the scaffold. And then if you load a protein, what is the loading efficiency? If the loading efficiency is very low, then it may not be very successful. So all these important factors play an important role when we talk about tissue regeneration based on cells, stem cells, and growth factors. And here where biomaterials play an important role. What are biomaterials? As per European Society for Biomaterial, they describe biomaterial is a material intended to interface with biological system to evaluate, treat, augment, or replace any tissue, organ, or function of the body. So this is a biomaterial. Biomaterial can be classified based on their source as well as based on their surface charge. So based on their source, we can divide them into natural source or synthetic source. In natural source, cationic means positively charged biomaterial are keratin, basic hydrolyzed gelatin, and then chitosan. These are the most popular natural derived material I listed out here. And then in case of anionic, we have fibrin, hyaluronic acid, alginate, collagen, heparin, also hyaluronic acid. Most popular natural biomaterials. And then in terms of synthetic, cationic cases, we have polyethyleneamine, cationic PAMM, polyl-lysine, and in terms of anionic, we have polyacrylamide, PLGA, polycaprolactone, PLLA. These are the most. In synthetic, we have non-ionic group, which is more important, which is called polyethylene glycol, which is non-ionic. Then let's discuss some of the most popular natural and synthetic biomaterial used in tissue regeneration. For me personally, the most important biomaterial is collagen, because collagen is abundant and isolated from the body of most of the tissues. So when we say collagen consists of three collagen molecule is made up of three polypeptide chain that consist of 1,000 amino acid. So these individual collagen molecule are helically coiled around by hydrogen bonds and covalent cross-linking to make a rigid rod-like collagen fibrils. So which hierarchically form collagen fibers. So if you see, this is the polypeptide chain cross-linked to hydrogen as well as covalent bonding to form the collagen fibril. These collagen molecule and then collagen fibril all together form collagen fibers, which is in less than a micrometer, very, very, very small structure. And then they form together hierarchically to form bigger organ. Like for example, bone consists 90% of the bone is consist of protein called collagen 1. So that's how that forms. So collagen is the most abundant protein in our body. As I said, they can be extracted from tissues. The novelty of collagen gel is their temperature sensitive gelation because of the teleopeptide. The teleopeptide molecule, what happens at neutral pH and at room temperature, they start to cross-link and become gel. So that is the beauty of this material. The fabrication of collagen gel we will discuss later. And then when we treat collagen in an acidic condition or a basic condition for hydrolysis, we can get gelatin. Gelatin, we are very aware of gelatin. All the gels basically made from gelatin, which is again very popular biomaterial. Then we have GAGs, glycosaminoglycan. So glycosaminoglycans are polysaccharides. These polysaccharide polymer recognized as heparin, hyaluronic acid, the most popular one, and then we have sulfates of chondroitin, keratin, and dermatan, as shown in the figure. So glycosaminoglycan are produced by mast cells of our body, and this also can be found in all type of tissues. Their interaction with a variety of protein helps to preserve these proteins' biological activity. In general, protein are positively charged. So glycosaminoglycan, because of the acid and sulfate, they are negatively charged. So in our body, when proteins secreted, these proteins are attracted by glycosaminoglycans in the matrices and sequestrating this protein, and they will release slowly whenever they are needed. So glycosaminoglycans are very wonderful. The most important glycosaminoglycan is hyaluronic acid. So hyaluronic acid. So hyaluronic acid consists of N-acetyl glucosamine and glucuronic acid. Because of this, it is a very anionic in nature. It forms hydrated matrices engaging in biologically important protein interaction. The predominant carboxylic group, the glucuronic acid, makes them negatively charged, so they attract positively charged protein. And then we have heparin. Heparin basically that participates, the blood clot. Heparin prevents the blood clot. So it is a highly sulfated glycosamine, consists of heterogeneous mixture of glucosamine and uronic acid or chloronic acid. The varying sulfation at each residue, makes this polymer highly anionic, so attractive for positive growth factors. In general, growth factors are also most of the growth factors are positively charged. So all these growth factors can be attracted by heparin and released whenever it's needed in our body. Because of this is highly used in tissue regeneration application. And then we have fibrinogen. Fibrinogen, as you all know, participates in blood clotting. It's a fibrous non-globular protein found in blood plasma. And then it has naturally as an abundant affinity for all these n number of growth factors, particularly VEGF-B, TGF-beta-1, BMP2, PDGF. VEGF and PDGF are the most popular growth factors for blood vessel formation. BMP2, BMP2 and 7 for bone regeneration. TGF-beta-1 for cartilage regeneration. So naturally, fibrin has a very strong affinity for this molecule. This fibrin is a dimeric glycoprotein composed of sets of 3 polypeptide chains, alpha, beta, and gamma. So fibrin gel can be prepared by polymerizing them with fibrin, with thrombin to yield fibrin monomer. Fibrin monomer forms fibrin photofibrils, which is upon branching makes fibrin gel. Fibrin gel also look like collagen gel and with a lot of porosity. So if we load a protein in this fibrin gel through this porous structure, growth factor is slowly released from the matrices. And then we have chitosan. So before we talk about chitosan, let's discuss about chitin. Chitin is the second most abundant biopolymer because it's obtained from the sea source, natural sources, particularly from the shell of crabs, shrimp, and prawn. And deacetylation, if we remove the acetyl group from chitin, we will get chitosan. Chitosan is highly insoluble in water and mostly soluble in acetic acid because of its crystalline structure. So what happens to make chitosan a water soluble chitosan, because water solubility is very, very important for your tissue regeneration application. So chitosan, upon functionalization of methyl group, which we call methylation, yield N prime methyl chitosan. The beauty of this material is it can be cross-linked with TPP, tripolyphosphate, for ionic gelation. That will yield trimethyl chitosan nanoparticle. This is called mucopolysaccharide, as I said before. That means it has a very strong adhesion in the mucosal area. So for for any mucosal tissue application, chitosan is the choice. And on top, pH less than 6.5 TMC nanoparticle, chitosan nanoparticle is soluble, and pH above 7, which is insoluble. This makes it attractive for the intestinal tract related studies because the pH in the stomach and the rest of the intestine changes. So this is very useful for such application. And the image shows how the nanoparticle of chitosan looks like. Because of its mucosal property, it's highly adherent. Then let's discuss about cell or tissue-derived ECM, which I used to work mostly on. So as we discussed before, cell-derived ECM can be isolated from the cell culture on a platform. And then we can collect the extracellular matrices as a thin layer or as a gel, and the structure of ECM pretty much look like collagen or fibrin gel, like a fibrous structure. That can be useful for the tissue engineering application. The beauty of this one is the complex and hierarchical extracellular matrix structure provides a microenvironment that is close to our native microenvironment. So it will be one of the potential biomaterial candidate for cell studies. So on top of this, ECM can be prepared from our own cells. So if we from the cells from fibroblasts obtained from our body, and then we can culture them, we can collect decellularized matrices from our own cells. And then alginate. Alginate is obtained from seaweed, sodium alginate, in the form of sodium alginate, and also from bacterial sources. It consists of hexuronic acid residues and manuronic acid. So this water-soluble polymer, the beauty of this water-soluble polymer is it is readily cross-linked with any cations, such as calcium and calcium as well as magnesium. Because of that property, it will be easily get any structure. So if we drop a solution of alginate on a calcium chloride solution, we get microcapsule of a beautiful microcapsule of alginate, and this is the SEM structure of the dried alginate microcapsule. And another important property of alginate is upon cross-linking, it has porosity of 5 nanometer. So biomolecules or cytokines will be slowly released through these pores and achieve sustained release over a period of 28 days. And then we can also control the release of growth factor in an on and off mode using ultrasound. So after we load proteins in alginate gel, if we apply ultrasound, these alginate gel releases protein in the presence of ultrasound. So we can apply ultrasound to release the protein and then off the ultrasound to retain to stop the release. So it has a beauty of that. One major disadvantage of alginate is they lack cell binding motif. So oftentimes it requires functionalization of cell binding molecules such as RGD peptide or other proteins like fibronectin or extracellular matrices. Then we have synthetic biomaterials. Two are the most popular synthetic biomaterial used in tissue engineering. Both of them received FDA approval for human application. So this is the structure of PLGA. This is the structure of caprolactone. Both of them are synthesized from using ring opening polymerization. It's a chemical reaction. So ring opening polymerization or polycondensation of cyclic lactide and glycolide will yield PLGA. PLGA is also an anionic polymer, and it mostly depends on what solvent we use to prepare them. And then PLGA can be pretty much used for any nanoparticle or microcapsule or scaffold synthesis. And this also behave like a biomaterial and achieves sustained slow release of protein upon degradation. The degradation of PLGA can be controlled by the molecular weight as well as the monomer ratio. So by changing lactic acid and glycolic acid repeat unit, we can make it slowly degradable or highly degradable, and also less elastic and more elastic just by changing the monomer. That's the beauty advantage of synthetic materials from synthetic prepared from a synthetic reaction. And then we have polycaprolactone, which is a linear aliphatic polyester made from ring opening polymerization of caprolactone. It's a thermoplastic and hydrophobic polymer, so which is extensively used in scaffold and in sutures and wound dressing, as well as other tissue engineering application for bone, cartilage, ligament regeneration. PCl is also anionic, has a low melting point and high viscoelastic property, which eases the production of variety of shapes and sizes, such as PCl pipe mesh, as we see from here in the picture. The last one in the synthetic biomaterial is polyethylene glycol. So polyethylene glycol is an inert hydrophilic polymer that consists of polyether groups which exist strong hydrogen bonds to water molecule. And PEG is available in different geometries and hydrophilicity and can be coupled to hydrophobic moieties to produce a non-ionic surfactant. The clinical viability of PEG is increased by surface modification with several functional groups like RGD, mono or diacrylate group, and such as and multi-arm PEG using vinyl group. Moreover, PEG can be easily combined with other biomaterials as well, like heparin can be combined, fibrin, PLGA or PVA can be combined with PEG to yield a composite biomaterial. The schematic shows here 4-arm PEG malamide microgel preparation using dithiophenol as a cross-linking agent. And we can use this biomaterial as well for growth factors and cells delivery in the body. And this is all about biomaterial. Some of the biomaterial I try to cover. We have so many other biomaterial, still new, different new techniques used to develop different advanced polymers for tissue regeneration application. But the one I presented here are the most studied and well-established polymers for tissue regeneration. Then let's discuss about some of the fabrication techniques. The first one is gelation. So collagen, we discussed about collagen. Collagen is is soluble in acidic condition. So in acidic condition, it will be liquid, and then this collagen can be neutralized using base such as sodium hydroxide, and then the concentration can be adjusted with media. Upon mixing thoroughly, load them on a plate, we can make collagen gel, like in any shape, like disc shape or cylindrical shape, it's like that. So this is a very standard temperature assisted collagen gel synthesis. This is how the collagen gel looks like after preparation. And the AFM image shows how the collagen fiber form the network on gelation. And then it can be also prepared as a particle and as well as electrospin fiber of collagen. And then the image so here is a collagen sponge. Collagen sponge or any hydrogel upon mixing, if we put in a mold and allow for lyophilization, which is otherwise called as freeze drying, all the water molecule will be evaporated and we can have a sponge. So this is very standard technique applied for tissue regeneration studies. And then emulsification. We discussed about nanoparticle synthesis in the earlier studies using emulsification. So emulsification is a very popular pharmaceutical technology being used to make emulsion. If we go to pharmacy, we see emulsion, which is like basically standard one. So in this case, a water or an oil base, so aqueous and aqueous phase as well as organic phase will be immiscible with each other. So using a surfactant and apply an external force such as mechanical agitation. In this case, we use ultrasound to collide aqueous phase and organic phase in the presence of surfactant to yield a particle-like structure. What we see here is PLGA nanoparticle, and on the top we have heparin microparticle, and this is hyaluronic acid microparticle. So hyaluronic acid do not have a cross-linking agent, so that means they cannot retain a surface a shape such as particle shape. So it has to be mixed with different polymers such as alginate to retain the structure of the particle. And similarly, heparin do not achieve in the shape, so which needs additional functionalization such as acrylamide. Methacrylamide is a temperature sensitive polymer. So at a room temperature, it will be liquid, and upon reducing the temperature less than 20 degrees Celsius, it will be solidified. So using this concept, we can make particles pretty much in synthetic and natural biomaterials. And then we have in fabrication method, we have electrodropping and spinning. Electrodropping is popularly used for making alginate microcapsule. So we earlier discussed alginate is a polymer, carbohydrate polymer, will be cross-linked with the help of calcium ions, and then that forms calcium alginate egg box structure. So using this concept, if we load sodium alginate in an electrodropping system, when we apply voltage like 7 to 10 kilovoltage, we can produce droplets, which will be collected in a collector containing calcium chloride cross-linking agent. Upon dropping the droplet in the calcium fluorosulfate, immediately it gets solidified, such as here. This is how it forms. And an advanced technique included in this system, adding a coaxial needle setup which contains inner and outer syringe with a spacer. By doing that, we can make core cell microcapsule. In this case, core is PLGA nanoparticle solution emulsion. The outer case is a cell is made up of alginate gel. And then we have electrospinning system. If we increase the electric voltage, applied voltage from 10 above 15, then instead of the droplet, it will be like a fiber formation that can be collected in the receiver to make scaffold. So here we see it's a standard scaffold used for bone tissue regeneration, made up of polycaprolactone scaffold. And then the last one in the fabrication method is microfluidics. Last before we have one more. So microfluidics are the very latest technology has been used for high throughput synthesis of microgel. So the beauty of microfluidics is like you can have multiple channel without any interaction until you want it. So in this case, basically it's basically an emulsification kind of method here. So an oil phase is oil phase will be guided through a different channel, and then PEG, in this case polyethylene glycol, mixed with protein or cells will be directed from a different channel. In a junction of channel, these both channel will meet with each other to make a droplet, and then that will be carried in a crosslinker to be crosslinked. By doing this, we can make microgels with cells or growth factor and also nanoparticle in microgel. This is how the typical chip, you can make the chip very small and simple, and this is the structure of a typical microfluidic channel used for microgel synthesis. Last advanced technology for fabrication method is 3D printing. So we have several type of 3D printing available. The one what we see here, it's called dynamic light scattering 3D printing. Basically, you can control the light source be exposed towards the top region where you will have light sensitive polymer. So in this case, we have gelatin functionalized with methacrylate. So methacrylate is a functional group, can be functionalized on gelatin, which we call gelma. So this gelma, upon come in contact with UV light at particular range, it will be cross-linked like this. So we can choose the size of the light exposure, and the shape of the light exposure can be controlled in 3D printing. So using this technique, we can make microgels of literally any shape and any size. What we see here is almost like 400 micrometers shaped star or square or star or square or flower like microgel. Hundreds to thousands of microgel can be prepared at the single run. So very popular technique used for the synthesis of microgel or growth factor as well as cell delivery. One of our recent paper is published in this year. It's in bioRxiv, so you can visit and check this work out. Then how to make porous delivery system. So whatever we discussed before, they are like for the generalized scaffold system or micro structure material system. But how do we introduce porosity artificially within the scaffold? By using either freeze drying technique or salt leaching technique. In case of freeze drying technique, in both cases we use porogen such as ice crystal for freeze drying mixed with polymer, and then we allow them to set in a mold, and then upon lyophilization, the water molecule will be evaporated, leaving the porous structure in the system. Similarly, in salt leaching technique, we use, for example, sodium bicarbonate salt with the polymer solution and then set in a mold make it as a gel, and then by dissolving, by leaving the scaffold in water, the sodium bicarbonate will be removed, will be dissolved, and leaving the porous structure. So this is very popular because it offers three dimensionality. So cells can be diffused, invaded into the core of the scaffold, and porosity significantly affect release kinetics of the growth factors. And with that, we come to a conclusion. Like smart systems are highly necessary for tissue regeneration application. So this chart is mostly used for designing growth factor delivery. So when we see, I have listed out cationic growth factors and anionic polymers, because growth factors should not be traditionally growth factor has been given as a short, and that causes poor clinical results. So we need a system that can be loaded with growth factor and delivered over a period of time until the tissue is regenerated. For an example, if a bone is injured, it takes almost a month to 3 months to heal fully. So we need a system that is preloaded with growth factor and slowly released within our body. So that means we need to have a kind of an interaction between the polymer biomaterial and the growth factor. So there is a strong interaction, but allow them to slowly release from the system. So to design such controlled, sustained delivery of growth factors, we can choose anionic polymers for cationic growth factors and anionic growth factors for cationic polymers. And then what are the target growth factor? We have to choose based on the literature. For an example, bone is highly been treated by BMP2, BMP7, and then combination of BMP2 and VEGF and PDGF are tissue growth factor. You can have like much more idea in the review article given here about that describes about all type of growth factor used for what tissue engineering application. At the preclinical level studies being listed out, all the biomaterials tested popularly, and delivery system in this one. And with that, I hope you guys enjoyed the lecture, and you come up whatever the question you have, please try to feel free to ask and then analyze through the chat. And I'd like to thank Dita for arranging this webinar. I hope this is helpful for everyone. Terima kasih. Thank you.
Okay, thank you, Ramesh. So for all the participants, if you have any question, you can raise your hand so I can unmute your mic, or you can write in the chat box. So actually, there is one question from a student. He asked about the stem cell for Parkinson treatment.
Parkinson treatment. Yeah. So far, if you look at the stem cell delivery, it has been highly successful for bone, and then we have been highly successful with cartilage. So when we say highly successful, that means so far the studies conducted in different field, bone, cartilage, muscle, cardiac, have been kind of getting positive results. And for Parkinson disease, yes, there are groups working on stem cells using neural stem cells for the repair of one. But the very detailed studies been going on, not very effective successful treatment so far identified.
Okay.
All the stem cells based can be potentially used for tissue regeneration, pretty much any kind of tissue regeneration. But understanding the phenomenon, the microenvironments that involves with the differentiation is quite challenging. So brain or anything inside brain, it's quite complicated. So we are at the level of bone, muscle and cartilage, but soon we will be there collectively,
Okay. So Fatur, understand. So there is another question from Alventa. And if our body reject those material that had been implanted to our body, how we fix this problem?
Yeah, that's a good question. So oftentimes, if you come up with a new material with no literature survey, then before we actually test in our body, we have to have a preclinical studies. So in our lab or in general, people who work with biomaterials are both natural or synthetic derived, they don't bring that directly into our body. So we conduct a lot of preclinical studies in mice or rats or monkeys, so to understand whether this material is safe, biocompatible, how much they are degraded, what is the immune response from a time dependent manner, for an example, chronological manner, a week after implantation, a month, 2 month, 3 month, and even 12 months, to check how the material is doing. So any material that doesn't perform well at the preclinical level will not be considered for future application. That means for clinical studies, it will be removed from the study.
Okay, can you get it, Alfonso? Is there any question from another student? I think Arinda is joining our lecture. Maybe Arinda, can you say something or want to ask something? So Arinda is from Universitas Indonesia.
Okay. I
Okay. So for another question, what do you think about where is the best choice between stem cell and autograft?
Yeah. So yeah, again, this is a good question. So when we talk about stem cells, imagine if we transplant, for an example, if we transplant a kidney from a different person to a different person, we have to suppress the immune system should be suppressed before transplantation. Same thing will happen as well. So that means when we are capable of deriving stem cells from our own body, then we can implant back to our body without compromising the immune system. So yes, you can get stem cells from any kind of animal like rat, pig, monkey, whatever, but applying them in our body may not be feasible or may not be advisable. So oftentimes, scientists, doctors, they collect stem cells from our own body, from the patient body itself, and then expand them, and then bring back to the patient. That's how the standard stem cell based therapy work.
Okay, I got it. So there's another question from a student, Kevin. So you want to ask about your opinion about the making a stem cell from the pig cell.
Oh yeah, I'm reading that question.
Yeah.
So Arinda asked about magnetic force change stiffness of cells.
Uh-huh.
It's mean in ECM scaffold we can add magnetic particle. Yes, that is very much possible. That was one of my idea during my PhD program to work on differentiation study, but I never did it. But it will be wonderful to see.
Okay. So a question from Kevin. He asked about your opinion about making a stem cell from the pig cell.
Yeah. He asked about making stem cells from the pixels, right? Yes, it is possible. Yeah, it is possible to study. To address that question, we literally can isolate stem cells from any animal or any human. But what happens? Stem cells obtained from individual, even from human being, if we collect stem cell from a 40-year-old and a 10-year-old or a 60, 70 years old, if you see, there will be so much variation. So we will not be able to chase behind this variation if we use stem cells from multiple sources. So what happens oftentimes in the field, researchers try to use one particular stem cells. So in our case, we use mesenchymal stem cells derived from bone marrow. And then oftentimes people use rat or mouse derived bone marrow stem cells because their genome somewhat close to human being. So that's the main reason they use rat derived stem cells and or mouse derived stem cells. But yes, it is possible stem cells can be isolated from any animal.
Okay. So there is one question from student. How about making pancreas from stem cell? And is it all of stem cell product affecting enough to solve our body problem?
I don't understand that question. Can you say me again?
So how about making pancreas from stem cell? And is it all of stem cell product affecting enough to solve our body problem?
Oh, that yeah. Actually, pancreas is it's a very delicate organ, and there is a group. If the student name is Diana, if
Yeah.
Diana is very interested about pancreas, there is this professor in Georgia Institute of Technology. His name is Garcia. He does a lot of pancreatic related study, and they use pancreatic cells, beta islet cells, for the implantation, and then understand how they secrete insulin to control it. Yes, it is possible. And then people are realizing the success story of pancreas regeneration. And we cannot say all stem cell product can be potentially solve our problem, but that is what we are working for, to understand how we can solve every problem. So far, not all the problems have been solved. So when first people doctors used BMP2, they were like, wow, this BMP2 is amazing, doing great job that regenerate bone than any other material does. But now we have problem with BMP2. BMP2 causes inflammation and also heterotopic ossification. So we need to identify what causes this problem. Maybe we need to reduce the dose and all the other things. So to that question, we are exploring this field little by little by little. So the more we know, the more we will be successful. So in my opinion, yes, it is possible someday that all the stem cell product potentially can address problems in our body.
Okay. And as far as I know that brain cell, I mean nerve cell in brain, it's not to the regenerative, right?
What is that as far as?
So as far as I know that brain cell,
Brain cell?
they cannot regeneration. So if the brain, so if something happens and damage the brain, we can do nothing.
Yeah, it's the most complicated organ thus far identified, because the micro environment that we discuss about. So when it comes to bone or cartilage, it's kind of easy because you can understand what is actually happening within the bone. Now scientists are at the level of understanding what is happening inside the brain, like exploring the mechanism. So when we have a mechanism of, okay, this is exactly what's happening in the brain, so we can now induce differentiation like neurogenesis. But so many neurogenesis study has been happening all around the world, and maybe down the line 5 to 10 years, we'll be talking about how brain can be regenerated as well. That's my hope.
Okay, so it means that in the future, stem cell could replace the brain cell. I mean,
yeah,
and it will be affect the function of the brain.
Definitely yes, because there are other factors involved in brain, right? The When we say brain has multiple components, and each components participate in different function of our body. So when we say what part of the brain we are intended to regenerate, and then that particular brain, if regenerated, will it carry the memory from the previous one? So there are a lot of questions involved with that. So we have to be you know we have to study more, do research more to understand neurogenesis field.
Okay, so there is one last question, I think, yes, from the student. So she asks about how about stem cell for replacing alveolus cells, for replacing alveoli.
What is that question? I don't get it.
So she asks about how about stem cell for replacing alveoli, pulmonary cells?
Pulmonary cells.
Yeah.
Oh, oh yeah. There are several studies that discuss about BMP4, for an example, growth factor BMP4 is being used for lung progenitor cell differentiation. So when we talk about tissue regeneration, like pulmonary regeneration or cardiac regeneration or any particular type of tissue regeneration, a group of scientists have been working on that. As far as I know, for lung is it's a very delicate organ. So people have been mostly working on the trachea, for the replacement of trachea, and not at the level of alveoli. But there must be, I may be wrong. There must be some groups working on pulmonary regeneration. As far as I know, people develop biometrical scaffold to replace as a replacement for trachea and stuff. But again, as I said, these are field. All the questions can be one day answered by exploring more in terms of research, and potentially can be done. So when we say for bone, I always quote bone because it is easy to understand. So when bone fracture, stem cells are recruited from the bone marrow itself. So bone marrow contains lots of stem cells that will be utilized for bone healing. Similarly, skin, underneath the skin, we have keratinocytes and other stem cells that participate in skin regeneration. So brain also have stem cells that will be specifically differentiate into neuronal cells. Similarly, alveoli cells potentially will be regenerated from the stem cells that resides in the alveolar niche. So we have to identify and then study to conduct a study on a bone or cartilage, which is easy in terms of preclinical model. But in terms of brain or lung, they will be quite complicated to conduct preclinical studies. First of all, the model itself will be hard to make, and then second of all, how how we will be performing the surgery, and then come up with the exact stem cells and scaffold for the implantation. All this makes complicated, but with the advancement in terms of 3D printing and then stem cell engineering, they all can be tested. So for those students who have a passion for research, they can start digging questions about this individual stuff. For an example, if the student wants to know more about lung regeneration, it's a very small field where you can explore more. Similarly, neurogenesis, many researchers have been actually actively studying about neurogenesis and brain repair, and then they can build the platform from now on to move towards such research.
Okay, so she asks more about stem cell therapy for COVID-19. What do you think?
What is that?
She asks more about stem cell therapy for
Uh-huh.
COVID-19.
For COVID-19, in case of COVID-19, we have been facing the problem to control the virus. So it's more or like it's like a war between our immune cells and the virus, right? So if the virus wins over the battle, we lose. If our immune cells fight over, we are successful. So the stem cell role in COVID-19 potentially can be bone marrow injection. For example, if someone's immune system is kind of low, that means they don't have enough immune cells to fight against this virus. So in that case, we can implant a bone marrow derived from a very healthy person into the bone, and then we can improve their immune system so that they can fight against COVID-19. So it's kind of a different route than stem cell approach, you know.
Uh, maybe her main focus is about stem cell to, as we know that COVID-19 attack our pulmonary tract. So
Yeah.
can stem cell therapy preventing our pulmonary tract or for treatment against COVID-19?
Potentially, I would say potentially difficult because stem cells will be like kind of an immature and undifferentiated precursor cells. That means they are like babies. You know, they don't have a they don't have a tool or techniques to fight against other. They are not prepared to fight in this competition. So what happens? We talk about hematopoietic stem cells, which I explained before. So hematopoietic stem cells, which is also a stem cell, that can be differentiated into monocytes and other blood cells, right? So if we induce the secretion of a potential immune cells that can target virus, but stem cell itself cannot target. Even if they encounter, that will be more or like a kind of weak cell to fight against the virus. In my opinion, I may be wrong. Yeah.
Okay, it's okay. So there's another question from a student about biomaterial for biomaterial to accelerating wound healing. Yeah. So she asked about what kind of biomaterial that can accelerate wound healing.
Yeah. So when we say wound healing in terms of skin, skin contain a lot of proteins, but the major protein is collagen. So depends on exactly what kind of injury, what kind of wound it is. We have multiple classification, level 1, level 2, level 3 kind of wound injuries. In a burn cases, it will be super hard because the entire microenvironment of the injury at the injury site is kind of collapsed. So it will be super hard to induce the repair or regeneration. But in terms of biomaterial, collagen, hyaluronic acid, or fibrin gel, they are quite popular biomaterial tested for wound healing. But these all are very standard biomaterials used for tissue regeneration, right? Recently, combination of materials has been prepared using 3D printing. So combination, when we talk about combination, we can say like Gelma with collagen, or PEGDA. It's a diacrylated polyethylene glycol with collagen. This kind of combination material can be potential for wound healing.
And what do you think about the amniotic membrane as wound healing?
Yeah, that is amniotic membrane, which we discuss about extracellular matrices derived from tissue or cells. So amniotic membrane, it's a potential material. It's been available commercially in the States in terms of different commercial product name being used for wound healing, even as well as for hard tissue regeneration, such as cartilage and bone and dental application.
Okay. So there is a question from Diana. Is it stem cell product can be suitable for baby? What kind of this product?
No, stem cell product, yeah, definitely can be suitable for whatever we apply for adults. can be also applied for babies. But when we say neonates and then babies, neonates, the immune system is kind of very weak. So the tissue regeneration ability is kind of low. But as we grow up, as baby grows up, the immune system, the stem cell has been will become very active. So whenever there is an injury, when we compare to young population and adult population, any injury that happens to young population tend to heal faster than adult population. But to that question, yes, it is possible. When we say what kind of product, all the extracellular matrices product, collagen, been in my review paper, I have listed out all the biomaterial oriented products available clinically for tissue regeneration application. All those can be used for kids as well, babies as well.
Okay. And I think it is the last question from Alfensa. As far that she know that tissue engineering can use living cell as building materials. For example, we can use fibroblasts for skin repairing treatment. Could you explain how we can multiply these living cells so we can use it for those treatments?
So yeah, that is exactly what we call expansion. So we get the cells, we isolate the cells from the body. For an example, let's say a fibroblast. So let's assume we have like a 100,000 cells or 1 million fibroblast is we have cultured on the plate. But when we think about application, we may need almost like trillions of cells for particular, depends on the wound site, for or for the depends on the implantation site. We may need a huge number of cells, so we have to expand these cells in the laboratory, in the tissue culture lab, expanding them, expanding them, expanding them. So depends on, some cells proliferate faster. For example, skin fibroblast proliferate super faster. So when we say super proliferation, if we put 100,000 cells in 4 to 5 days in a expansion or growth media, it will become 1 million cells at the end of fifth day. So if you have 1 million cells at the end of fifth day, if you culture them into 100,000 cells into 5 plates or 10 plates, you will have 100 million cells at the end of 2 to 3 weeks. That's how we expand and then bring back to the application.
Okay, one more question from Kevin.
Uh-huh.
Is it stem cell product can be growth with the growth of the body cells? And is it connect to the another neuron?
Oh yeah. So when we talk about the cell or product, when we use them, there will be an orchestrated and then combinatorial effect. So if we use growth factor, they will not only promote proliferation of one particular cell. They will, in general, have multiple functions on different type of cells. And then to the question, um, if a neuron is differentiated, and obviously that neuron start to explore to repair based on what condition they live. If there are enough cells, they will just stop proliferating and start exhibiting their function. If in order to repair the gap or the injury, they start to proliferate a lot just to focus on wound healing. So it's more like a collective orchestrated mechanism that participate in tissue repair. So every time when we bring a product into the body, and then we can expect a collective approach to accelerate repair.
Okay, thank you. So I think it's
Thank you.
enough for the discussion today. So uh, we can conclude that tissue engineering is the promising solution for the medical problems, especially to replacing damaged organ and to to build the to build the stem cell as we want. We need to with an appropriate environment. Is it right?
Mm-hmm.
Yeah. So we can define the cell fate based on the microenvironment, so they can differentiate as we want, right?
Yeah.
Okay.
Yeah.
So I think because before I close this this lecture, I want to all students to activate the video so we can take a photo together for the documentation, I think. Wait a minute. I'm gonna.
Uh-huh.
The participant, could you turn on your video? Okay, if they cannot do so. Thank you for your time, Mr. Reyes.
Yeah.
Yeah.
Yeah. Thank you. Thank you. Thank you. All the best, to all the students, and let's be in touch. If they have any more questions, just let them know that I'll be available to address the questions anytime.
And maybe you can say something in Korea because most of them are fans of Korea.
Oh yeah, maybe we can have a separate discussion someday. And next time about Korea, or like we can have a seminar on how to prepare to go to Korea or to go to foreign international universities. That potentially will be helpful.
Okay, so thank you for today and thank you for all the participants. Okay,
Yeah.
I'm gonna
Thank you.
end the meeting.
Terima kasih.
Okay,
Yeah. Right.
sama-sama.
Bye.
Bye.